why we're different from one another. Something is wrong with the slides. Okay, yeah. And why is this important? So everyone is trying to move medicine towards personalized medicine or precision medicine, where we are not trying to treat the patient rather than treating the disease. But to do this, we need to know what makes each patient unique. And to know what it makes each patient unique, we need to try to understand the different things that define each patient, each person, what makes each person unique. So how do we do this? Or can we think about things that we could look at? What would a doctor want to know from us? There are things like, genetic ancestry, for example, or sex differences, age, our weight, behavioral, like diet, running, smoking or drinking, and our clinical conditions. And what we do to address this question is we look at the... Sorry. It does. No, I think it does. Yeah. What we do to address this question is we look at what we call the human transcriptome. So everyone knows what the DNA is. All our cells in our body have the same genome, and then we have around 50,000 genes, and each gene is going to encode a protein. And what we want to know is how each gene is activated in each of our cells in our body. And this changes as we age, this changes as when we are sick, this changes whether we are a female or a male, this changes throughout our life and can explain the question that I posed, why are we different from one another? And this is like the readout that we're going to use in our lab and in a lot of our talk. Basically, it's a readout of how each gene on our genome is expressed in every cell of our body. And how do we do this? Well, we use massive sequencing of the transcriptome, and this generates a huge amount of data. And then this is the idea that I was mentioning before. All our cells have the same DNA, the same genome, but what defines what each cell is going to become at every point in our life is the transcriptome, how these genes are activated in every time of our lives. So all organs have the same genome but different phenotypes, and the same thing as we age, we have the same genome, but the genes that are expressed or activated throughout our life are different. And what do we need to study this? Well, we need a high-performance computing. We need a supercomputer because we have, like I said, 50,000 genes and a lot of cells in our body, many patients or healthy donors that we need to look at. So we need a supercomputer to analyze this data. This is Mare Nostrum 4, which was inside a church, and it won several prizes because it's known to be the most beautiful supercomputer. And this is Mare Nostrum 5, which is in a new building at the bottom floor of the building, and you can come and visit to see it. So basically, what we do in the lab is to try to go from gene expression to phenotype. It can be healthy phenotypes, like why differences between males and females, or aging, but also it can be why do we get sick. Okay, so I'm going to tell you about a project where we asked the following question. If we look at the gene expression, gene activation, through many tissues in our body, which tissue or organ do you think it changes the most as we age? Or which organ do you think is the most different between males and females? And also, which organ do you think might be most different in BMI? And pick or between individuals of different genetic ancestry, for example, European and African individuals. Pick one individual trait and think about which organ do you think maybe changed the most with age, for example. Does anyone want to guess? The organ that might change more. Or if we look at males and females, what organ that we both share, right, because there are organs that only males or only females have, is going to be the most different between males and females. And the same for ancestry. And I was really interested in this question because this, of course, has implications for what I was telling you before, for precision medicine, right? If we need to treat an organ that changes a lot with age, and we have patients of different ages, we need to understand why this is happening. Okay, so to address this question, we used a data set that is called the Genotype Tissue Expression Project, where we have data from 1,000 individuals, 46 tissues, and 20,000 samples. And for all of these samples, we have information about the transcriptome, about the activation of these 50,000 genes that I've told you about. And then we can try to understand, this is the distribution of the different phenotypes. So in ancestry, we have African ancestry individuals and European, the distribution of male and female, the age distribution, which is centered around 50-something years, and BMI. All right, and then we model gene expression. I don't want to go into details of this, but basically this is the idea. For every organ, we try to address how age, sex, ancestry, and BMI contribute to this gene expression variation. And here is the table that answers the question that I put before, where we can see that, for example, the tissue that changes the most with genetic ancestry is the skin, okay? And this makes sense because we're comparing European and African individuals. And this is, of course, when you think about it, this is very obvious. The skin is going to be different. But what makes this project special is that we have all of the genes now that explain these differences. So we're going to phenotype to understanding the molecular basis of these differences between individuals. In sex, the tissue that is most different is breast, because in females, breast has a function, which is lactation, and in males, it doesn't. So of course, this makes a lot of sense. But again, we have the genes that explains this difference, the molecular explanation why male breast and female breast are different. And in aging, we see that the arteries are the tissues, these are the two arteries, that have the largest difference. And again, this was already known. Oh, sorry. There is this medical doctor that said, a man is as old as his arteries. So if everyone knew that the arteries are aging faster than other tissues in our body. And in fact, cardiovascular diseases are the main cause of death worldwide. But we have the genes that explain these differences. And in BMI, this is also expected. We have the adipose tissue as the tissue that changes the most with BMI. And this work is already published. And it had a lot of, the media was very interested because it said that it was explaining what makes each person unique. I mean, we're not explaining what makes each person unique really, but we kind of are starting to understand all these demographic traits and the individual traits, how they are molecularly explained by these gene expression differences. And then we also wanted to know if there are clinical traits that explain these differences. And in this same database, we had information about certain conditions like diabetes, smoking, and different other conditions. And one of the ones that we wanted to focus on is diabetes, because diabetes is highly prevalent. So it's a chronic disease that occurs, and it has a high prevalence. It's 1 out of 9 patients, I think, that get diabetes at some point in their life. And there is 2 types, type 1, where the pancreas does not produce enough insulin, or type 2, where the body cannot effectively use the insulin that it produces. And insulin, as you probably know, is the hormone that regulates blood glucose. And the question that we ask now is, okay, is diabetes impacting only the pancreas or the adipose tissue, or has an impact in the other organs? In other words, it's a similar question than the one we posed before. Are all the other organs affected by having this disease condition? And we did a similar analysis. Okay, the question is type 1 and type 2 diabetes, do they have a systemic impact on the transcriptome? And can we find the genes that explain this impact? And here I'm showing a table where we have the tissues again and the number of differentially expressed genes, or the genes that are different between cases and controls. And what we see is that there are many genes that are differentially expressed between diabetic patients and not diabetic patients. So diabetes has an impact in many organs of the individuals. And the second thing that we saw is that actually the nerve was one of the tissues most affected. And when we started the project, we didn't have a lot of knowledge about diabetes. So I was skeptical about this finding. Many things can happen when we use this kind of large-scale data analysis. There could be batch effects on God knows what. But what was interesting, though, is that both type 1 and type 2 diabetes showed signal in the nerve, suggesting that this could be real. And then in this project, we also have histological images from the same patients. So the hypothesis was, okay, if the nerves are really affected in these patients, maybe if we look at the images, we see differences between the cases and controls. And then we started to use deep learning, because now, of course, is revolutionizing imaging analysis. And what we did was to train a simple classifier to try to see if the classifier could separate between cases and controls. I skip one slide. Okay. And what we found is that the classifier had 76% accuracy, classifying diabetic patients versus non-diabetic patients. So something really was going on in the nerve of diabetic patients. And you can see here an image of healthy and diabetic patients. So the images really look different. And when we started reading about this, it turns out that there is a condition that is called diabetic neuropathy that affects 1 out of 2 patients at some point in their life, which causes pain, and it's driven by this accumulation of glucose in the nerves of the patients. And it can even lead, in the worst cases, if untreated, to amputation of the patient. So it's a condition that exists, and there is no active treatment for this. There is no cure for this disease. There's only a treatment a posteriori. And then, okay, we asked, can we then use these classifiers to actually find the genes that might be explaining this phenotype that we see in the patients in the nerve? And what we did was to take the probability of being classified as a diabetic patient. And now we're not separating between yes or no. We're using the probability as a proxy for severity of the disease, and then try to find genes that are correlated with this probability. And what we found is that there were 328 genes that were correlated with the probability of being diabetic in the images. And, sorry, a few of them were already known, but most of them were novel. And what this means is that these genes are new molecular or new potential targets for treating this disease. And sorry, this work is carried out by José Miguel Ramírez in the lab. And then, of course, it's not only important to understand how how individual traits like aging or sex affect us, but also what do we do, where do we live, whether we exercise or not, whether we smoke or not, or whether we drink or not. So our behavior also has an impact on the transcriptome. And so to start tackling this, how our behavior can affect molecularly our gene expression, we started a project where we looked at the transcriptional and epigenetic impact of cigarette smoking across human tissues. And again, we were using the same data set where we look at many tissues, well, the same tissues that I've been talking about, and this activation or deactivation of the genes. And we asked the question, does smoking affect, apart from the lung, which everyone knows, and you've seen these horrifying pictures, does it have an impact in other organs, in other tissues? And can we measure it, and can we find the genes affected? How does cigarette smoking impact the transcriptome? Okay, so we ran this analysis that we do, we ran differential gene expression analysis, and what we saw is, of course, the lung is the tissue that is most affected, but we find genes in almost all of the tissues. And actually, these tissues where we find no signal this is the sample size. So the tissues where we find no signal are the tissues with the lowest sample size, suggesting that maybe it's a power issue, but that the smoking does indeed affect most of, systemically, all of our organs. The next thing that we saw is that thyroid is the second most affected tissue. And again, now we're also going to use the images to address whether this is a real result or not. So we did the same thing. We used transfer learning on a convolutional neural network to train the last layer and to classify between smokers and non-smokers. And the classifier, I don't know if I put the accuracy. Yeah, the accuracy was very good in lung, but also in the thyroid, suggesting that the signal or what is happening in the thyroid of the smokers is real and is not an artifact of this data set. And again, if we look at the never smokers, this is, I think, is the lung, yes. In the never smokers and the smokers, the difference is that we think that the deep learning model sees is that there are macrophages in the smokers, so that means that there is a lot of inflammation in the lung. And in the thyroid, the glands of the thyroid are much bigger in the smokers compared to the never smokers. And then, of course, this is, that deep learning is a black box, but that was our guess by looking at the images that were classified with the highest probability and the lowest. But if you actually measure here, it's supposed to say size of the diameter of the gland in pixels. And what we can see here is that if you measure the diameter of the holes that we were seeing in the previous image, smokers have higher diameter. So actually the glands of the thyroid on smokers are much larger. And then, okay, is there hope? If we stop smoking, what happens? And what we did was to then train a classifier on the ex-smokers. Sorry, use the classifier trained on ex-smokers and non-smokers and see how the ex-smokers were classified, okay? And if the ex-smokers go fast to baseline levels, they should all be classified as non-smokers. If the ex-smokers take a lot of time to go back or never go back, they would be classified as smokers. And what we see is that there is an intermediate answer. And so if you stop smoking, you can get to baseline levels, because here we have many. I don't know why there is missing information in this slide, sorry. It appears now, okay. So the never smokers, so here is for gene expression, sorry, and the never smokers, sorry, so 75% of the ex-smokers are classified as never smokers. So what this means is that if you stop smoking, you can go back to baseline levels. And for the smokers, some of them are classified as smokers, so they still have not gotten to baseline levels. So there is hope. When you look at gene expression, when you look at methylation, which is a modification in the DNA, it's a different type of omic, but it's basically the smoke can affect the DNA directly and modify it. This takes longer to go back to baseline levels. So the answer is, can you get rid of the impact of smoking? Yes, but the earlier you quit, the better, because gene expression is going to go fast to baseline levels or faster, but DNA methylation, which is a modification of your DNA, might not go as fast. And the other thing that we saw is that there's been several papers that have pointed out that smoking has similar effects than aging. And we wanted to see if these were true, first of all, and if this is true in all of the tissues. So what we did was to overlap the genes that are differentially activated between smokers and non-smokers, and the genes that are differentially expressed or activated between when you age. And what we saw here, I'm showing, here are the overlaps, and when it's red, they are going in the same direction. So up when you age and up when you smoke, and in blue, in opposite directions. And what is very clear, right, is that it's not 50-50, there's a bias. And this suggests that if you smoke, your tissues are aging faster than if you don't smoke. And this shows that this is happening at the molecular level. This is the preprint. This is work by Jose Miguel as well. So if you're more interested, you can find the preprint. I mean, this is going to be published very soon in Genome Medicine, I think maybe next week, but the preprint is already out, so you can take a look at it. And I want to go back to this question that I asked, that which tissue changes the most as we age, and the sentence that this guy said, a man is as old as his arteries. He actually was a medical doctor. And this sentence, well, he was right, in a way, but not entirely. And as you know, for many years, medical doctors in medicine in general have taken men as their reference, and they've been always looking at how men behave, how disease affected men, how can we treat using men or rats, or male rats. So is this true for women, or is it not true for women? And actually, arteries in females age a bit differently than those of men, but if you split between men and women, the arteries also age very fast, in women too. But there is one tissue that ages faster even, and is here, the uterus, okay? Because if you look, this is the sample size. The sample size is extremely small of the uterus, and we still see a huge signal of aging in these uteruses, and the same in the ovaries. And for us, this was very interesting because in females, it looks like arteries might not be the tissue that ages the fastest, but actually it's the uterus. So we wanted to dig in a bit further on this, and we thought, okay, maybe this is driven by menopause, which is the time when females stop being capable of reproduction and they stop cycling. And we thought, okay, is menopause driving the large changes in gene expression? And what we did was to look at the differential gene expression, but in age bins, to see if the differences that we were seeing occurred specifically when you compared females that were between 30 and 40, 40 to 50, or 40 to 50 to 50 to 60. And the natural age of menopause is 50. And what we can see here is that the number of genes that change expression is really happening at 50. So it really was strong evidence that this was driven by menopause. If we look at the trajectory of these genes, how they change, some genes go up in expression around 50, and some genes go down in expression around 50. So that was pretty clear for us. But then, when we were looking at the function, of these genes, I was thinking, okay, if we look at the function of the genes, they are obviously going to be annotated as being important for menopause. And we looked at the most common database where the function of the genes are there. And we saw that there was no function for these genes annotated related to menopause. And I was very surprised. How is this possible? And then we said, okay, is menopause even annotated as a functional term in these databases? And it turns out that if you look for puberty, which is the opposite process, is the start of the reproductive life, but this includes males and females. There are 3,000 genes annotated. For female puberty only, there are 32. And for menopause, there is 1 gene. And this really pushed us to try to address this by really focusing a project on trying to understand the genes that are explaining the changes that females go under menopause. And this, for me, was crazy because all females in the planet are going to go through this. So how is a process that is going to happen in half of the population not have any genes annotated? This is really getting at the bias that has been there for a long time in personalized medicine. No, that males have been taken as the gold standard. Okay, so what we did was to try to address the histological and molecular dynamics of human-female reproductive aging. We have breast tissue and different parts of the female organs. These are the ovaries, and these are the uterus. For those that are not familiar with this, the uterus is where the baby is going to be when we get pregnant. And the ovaries are basically the gonads, where the gametes are created. And for this project, we directly use the images using deep learning and the gene expression, or the RNA. And we have information about different organs from up to 300 female donors. The first thing is, of course, menopause was not annotated in this database either, so we didn't even know if females had undergone menopause or not. So we tried to use the histological images to annotate this. And what we did was to train a convolutional neural network, again, using transfer learning to train the last layer and get a prediction at the tie level and then at the donor level. And the accuracies were quite high for uterus, ovary, and vagina, and not so much for breast. And then, when we looked at the probability distribution, we also had an interesting finding. And this is that the uterus, which is this one, light blue here, had a huge switch in change in the histological images, suggesting that the uterus changes a lot at around 50 during menopause, but the ovaries start aging much earlier. If we look at if we look at gene expression, the same thing happens. So we have the uterus being transitioning around 50 and the ovary transitioning much earlier. And this makes a lot of sense, again, because fertility decay starts around 30 or 35, but menopause occurs much later in life, around 50. And again, this is important because we can identify the genes that are driving these two processes, and they are decoupled. So if we now look at the probability of being old in the uterus and the probability of being old in the ovary, what we see is that we have many females, most of them, that have an old ovary, but a young uterus. And I mean, of course, these are females around 35 to 40 to 50-year-olds. Okay. And then I've been using tissues and organs independently, but they're not the same thing. An organ would be, for example, the heart or the uterus, and the tissue would be the different parts of the organ. And now I had the question, okay, if this aging is asynchronous, so ovary ages earlier than uterus, can we see this at the tissue level? Maybe the different tissues within the organs age at different paces. And there's a lot of debate in the aging field whether aging is a linear process or it happens in bursts. And in fact, it makes a lot of sense that maybe aging, I mean, we don't age at the same pace, but maybe our hearts age faster. And if this happens, we might die of heart disease. So for me, this was an interesting question, not only to address human reproductive aging in females, but in general, in the aging field, can we show that the different tissues age at different rates? And for this, we use vision transformers, where you can actually use tiles that are cut in smaller pieces and do a similar thing, train a classifier of these tiles to address whether different tissues or small pieces of the organ age at different rates. This is a UMAP where we can see that the different tiles are clustered similar depending on the tissue they belong to. This is the uterus, so these are the different tissues. And we see that they are clustered similarly. And if we then look at the trajectories, these are the two tissues that we could classify or look at in the uterus. And what we see is the myometrium and vessels transition at 50. But in the vagina, for example, this is different. So in vagina, most of the tissues, lamina propria and stroma, age linearly, but the epithelium has this transition. So using the images analysis, what we see is that the aging in females is tissue-specific. And we cannot generalize, you know, the uterus gets old or the ovaries get old, vagina gets old at the same rate, no. And this is likely happening in all of our organs. Aging might not be happening at the same rate linearly, but specifically, every cell type might have the different dynamics. And then, okay, the question is, can we find the genes driving these differences and these changes at the tissue level? And basically what we did was to use an early integration method to try to find whether we could find genes that were associated with changes in the images for particular tissues and were related or correlated with gene expression changes with age. And here I'm showing the results where we have the different factors which are latent variables that explain variability of changes in the images. These are image changes on gene expression. And what we see is the most correlated factor with age explain both gene expression and epithelium histology variation, which is here. This factor changes with epithelium in vagina and with gene expression. And this means that pathways associated with epithelium development, skin development, epidermis, are the genes that explain these changes in the vagina. And this is again important because we've known for a long time that epithelium gets thinner in females with menopause, but we don't know which genes were driving these changes. His work is a preprint. It's under review right now. And this is work by Laura Ventura and Alexandra, a postdoc in the lab. If I go back, it works. It's crazy. Okay. So I've mentioned that females and males age differently. And a lot of the work that I explained to you today is by looking at gene expression of the full transcriptome. So we have gene expression data for organs altogether. But I think it would be much better if we could look at gene expression variation in single cells. For example, so what I've been showing you so far is gene expression in the uterus, in the heart, in the arteries, like a fruit salad, no? All of the cells are mixed into in the same fruit salad, and we do kind of a smoothie, okay? But ideally, what we would want is to have the gene expression of every cell in our tissue. And now the technology is ready. There is this technology that is called single-cell RNA sequencing that allows to have the gene expression of these 50,000 genes. It's a bit less than that in single cell, but 20,000 genes, let's say, in every cell in our body and for many, many donors. So we started to think about, can we use this technology to study aging? And one of the projects that we focus on is to study sex differences in the immune aging at single-cell resolution. And I said before that males and females age differently, but at that time, when we started this project, there was no project so far that had really looked at how the immune system ages differently between males and females. And we decided to address this by looking at the database that has information about 1,000 donors, 50 females, 50 males. And I think this is very relevant because males respond worse to vaccination and to infection, especially with age, whereas females respond better to both vaccination and infection, but also we tend to have more autoimmune diseases. And the basic for this is not well understood. So we thought that maybe we could start shedding light into these questions by looking at how gene expression changed at single-cell resolution across all of the immune cell types that are there. Oh yeah, I said that. Are there sex-specific cellular composition changes with age? So here we do a different approach. We don't look at how genes change, but rather we try to classify cells or cluster cells based on their transcriptome. So we take all of the genes and their activation for every cell, and we try to find similarity metrics to see how these cells are similar to one another, and then how they change with age, and then answer the question, are these changes in cellular composition different between males and females? And these are the results. So basically, what I'm showing here is the log-fold change of these abundant changes of the different immune cell types. I don't expect you to know what these immune cell types, but these are different cells of the immune system because the immune system is super complex. But basically, what I want you to focus on, these are females, these are males, and the general trend in this plot is that, with the exception of these B cells, females have more colored cells. And these colored, well, these are groups of cells, really. We call them neighborhoods, groups of cells that are transcriptionally similar. Females have more neighborhoods that are colored. And that means that they change expression and abundance with age. So in general, look here, for example, in monocytes, here in this other type of monocytes, in CD4, in TCM. I mean, the signal is much stronger in females than in males. So what it means is the immune system in females is changing much faster with age compared to males. And then we wanted to zoom in and look at these particular cell types a bit in more detail to see if these groups of cells had any specific function. Are they all behaving the same, or do they have a function that we can understand and can explain these differences in autoimmunity or response to infection and so on and so forth? And the same for these B cells that are this group of neighborhoods of cells that are changing so much with age in males. I'm going to just give you two examples. I'm going to give you the CD8 T effector memory cells and the B cells. What are these cells doing? What we do to address this is we look at the gene expression again, and we look at what's annotated for the genes that change expression in these cells. And in CD8 T effector memory, what we saw is that the genes that, well, we do marker gene identification to identify the pathways that are enriched in these cells and functional enrichments. And basically, what we saw is that CD8 T effector memory cells, which are cells that are fast. I mean, you need to imagine that these CD8 T cells are in the tissues, and they're waiting for pathogens or cancer cells to attack. And these are the ones that would remember these infections and attack them. So they are fast, local, and specialized responders to attack infected cells. And what we see when we look at the enrichments is that there is these cells that change with age, specifically in females, are associated to a specific type of cells that are called cytotoxic. So they are even more reactive to infections and more reactive to pathogens. This is called cytotoxic, which means that they are attacking and killing the cells that are infected or the cancer cells. So in females, these cytotoxic cells are more abundant. And what's more important even is that these cells that have this marker gene doesn't matter, but these subpopulations have been associated to severe autoimmune diseases. So I guess what I'm trying to say is that in females, we already see that the abundance of specific immune cells that have been associated with autoimmune disease increase in abundance more in male as we age. And here I'm showing, again, the plot that I showed before, because we also wanted to understand what is going on with these B cells. These have the largest log-fold change, and they only happen in males, which is very intriguing. And the B cells are the cells that produce antibodies. So again, these are very important to fight pathogens. And what B cells do is that when they have antibodies to fight a particular pathogen, and when this pathogen re-enters, these cells start expanding. And so sometimes these expansions happen in cancer, even though the pathogen is not there. And what we saw is that this particular B cell or population has a marker that is CD5+, and it's been shown that having an expansion of these clones increase the risk of developing a B cell-related cancers like chronic lymphocytic leukemia, which is a type of blood cancer, and these type of cancers are more prevalent in males than in females. So we kind of are linking back our observations in healthy aging. These are healthy donors, they don't have cancer. But we are picking up on something that is happening earlier in these donors that can explain later vulnerabilities that are sex-specific. So females have larger aging effects in most of the cell types, but not all, both in cellular remodeling and in transcription. And we also see that there are sex-biased expansion of subpopulations that are hallmarks of immunosenescence, which is really the aging of the immune system, related to higher inflammation and autoimmunity in females, and to B-cell leukemia in males. And again, this is a preprint that is currently under review, and this work is led by Aida Ripoll, see if I can get the name, okay, Maria Sopena, in my lab. And I want to finish my talk just by saying that aging is only half of the story. I mean, we've been looking all the time at adult donors, but there's, maybe 1/3 of the population are not adults, are kids. And many things of what is happening to us as we get older might have an origin when we're young. But the problem is that working with kids, of course, is very challenging. It's very hard for obvious reasons, no? I mean, ethical reasons, to get tissues, to get blood, and it's very complex. But I'm very happy to be part of a project that is really trying to address this, that is called Developmental GTEx, where we will have data and information for different age span, going from 0 to 18-year-olds, for all tissues and gene expression on healthy kids through development. And this is fundamental, because if we want to understand, what happens when we get sick, we really need to understand what is the baseline when we are healthy, how these genes and these molecular layers are when we are healthy, and this is extremely challenging to have information about this, and we will tackle this by studying how development happens in small children. And with this, I want to thank my lab. This is an amazing lab that, I only mentioned the first authors of each of the work, but it's extremely collaborative, and pretty much almost everybody participates to all of the projects in the lab, so I want to thank them. My collaborators, all of the consortium that I'm involved in, and the funding, and you for your attention. Thank you very much.
Thank you very much. Are there any questions? Well, you think about questions is like coming back to the last part that you said about the immune system. So it seems like your immune system, when you age, becomes more active.
Yes, in females.
Yeah,
in general.
Partly in males, but in a different way.
Yeah,
but in
totally,
general, yes. Why would that be? Because in the end, it has to be an evolutionary advantage. What would be the advantage?
So, it becomes more active in terms of inflammation. So a lot of the aging community thinks that inflammation so it's very important to activate the immune system when you have a pathogen, and it's very important to turn it off. Because you need to go back to baseline level, because you are damaging the tissues if you stay, active for a long time. And I think in aging, what happens is the immune system doesn't work that well, and you don't you're not able to get it back to baseline levels. So you have this state of inflammation that also happens in smokers, that doesn't get the tissue to be healthy, to go back to health levels, to baseline levels. And this is not an adaptation, it's, let's say, a malfunction of the immune system that gets active and doesn't go back. And autoimmunity is a bit the same thing, right? In females, we are better of responding to pathogens, but we are not so good at turning off the immune system. So
but how would this evolutionarily have come? That's a little bit weird, no? It's like...
evolution cares until we reproduce. And then after we reproduce, evolution doesn't care. Unless
you say that the grandparents are taking care of the kids and they survive better.
Only for females. But this is not, right? You're right. But this has not been shown. The menopause, this is the hypothesis why menopause happens, because ideally, you would want to reproduce for a long time. And not many species have menopause, and I agree. But in general, right? In general, whatever happens, and this is the theory of aging, right, is that evolution won't care whatever happens to you after you reproduce. So then, that the immune system malfunctions after you reproduce doesn't have any consequence, evolutionarily speaking. You want to have a good immune system until you reproduce, and then, yes.
Questions by now?
Thank you. Very impressive presentation. Thanks a lot. I was wondering more on the methods, like on the methodological side, what sort of tools you use for the analysis, because it looks like it's super complex. You have temporal dimension, you have all these factors, age, sex, a lot of components. And sometimes I guess it must be quite challenging to actually identify some sort of trajectory across this super multidimensional space, right? I see you use UMaps and you use other things, but are there any things that you think we need to work more on the methodological side to bring, more power of analysis that you think is now missing? Or?
So the limitation for us is not the computational power, although it might be a limitation for many people. Right now, for me, the limitation is that methodologies to identify non-linear trajectories are not really good, and the approaches that people use in the field are extremely simple. So in the lab, we're really obsessed by trying to develop new methods, and I haven't even thought exactly how we're going to do this, because now we use these classifiers and infer the trajectories, but maybe we should kind of get a way of doing inference and putting a p-value on these non-linear trajectories, and we've done it, but I'm not entirely sure that this is the right way to do it. So I think, and this is a huge question, again, in the aging community. It's not just me trying to find non-linear trajectories. There's been several papers suggesting that aging is not a linear process, and we need to find better methods to really address this. So for me, the limitation should be on trying to develop better tools, this is from the transcriptomics part, on identifying nonlinear trajectories, yes. And then, of course, another limitation is that not a limitation, but the more donors we have and the more data we have, for large cohorts, we can understand what is happening in healthy conditions, which in many cases is harder to get. So I think, both data and models are needed to tackle the next questions. Yes.
Thank you.
Yeah, I have one. Yeah, Marta, thank you. Thank you very much. So I'm wondering whether you then, so I have a couple of questions actually. So first one, I'm wondering whether you can then cross this data with data on epigenetic changes that are known to happen with age. So there are quite a number of people working on that.
Yeah.
And then the second question is whether you have then the ability to enrich these transcriptomics data with ontologies in order to be a little bit more explainable in terms of biological functions. For example, one question that popped up in my mind is how the trajectories you're seeing might be related with the evolution of hormone receptors, sensitivity to specific hormones that change a lot with age and are strongly sex dependent too.
Yeah, so I agree. I mean, ideally, we would want to have, for all these donors, I mean, the more information, the better. Epigenetics would be great. We don't have it for these large cohorts, and this is a limitation. We've been able to look at methylation in some instances, which is these changes in DNA. But most of the samples in these large cohorts and the type of analysis that we do, we don't have epigenetic information for all of the donors. But I think, yes, because then if you have the epigenetic layer, you don't only see the genes that are active, but you see how they are regulated. And this could be extremely informative to understand the biological mechanisms that explain this. And the other question related to hormones, yes, I mean, for me, in these developmental GTEx, in the development of kids, one of the things that I want to do is to get measures of the hormones, because a lot of the things that we see probably are explained by hormones. And because we're looking at this from a systemic view, from the full organism, all the organs, having the transmitters, which are the hormones really that go throughout the body, is fundamental. But this information is not there. Nobody cares about the hormones. I mean, in the community, nobody cares. I mean, there's no information.
Me to another question. Is about Marta. I love all your talks, so I tell you every time, but to this audience. But this brings me to a question of gender.
Huh.
Like, if you are thinking hormones, then gender starts to play, because then people are taking hormonal therapy, or that, for example, smoking can also be related to a gender condition. So how do you put gender in this? Do you have a sample size for non-binary?
Yeah.
Are you thinking about this, or are you doing something about it?
That would be great. So just to put in the perspective Paula's question, so sex relates to the biology, right? What chromosomes you have. And I don't know if you mentioned this in your talk, or no, the gender. Okay, no. And then gender, right, yeah. And then gender is a choice and relates to probably the behavior, right? So you choose what gender you want to be. And so far, you're right, we've only looked at sex, biological sex, and I think it would be extremely interesting. In fact, I was reading a paper yesterday of people that have had immune changes in trans donors, and this is an statistic thing that is great, because the chromosomes are from one sex, and then the hormones are from the other, because they hormonate them, and then you can see what is driven by which. For us, the sample sizes are not still big enough, right, to find people that is trans in these cohorts, but I think they could be extremely informative, and also we don't have information about self-reported gender in this particular cohort, but I think it would be, and also when you have a mismatch, what happens is people discard them, because it could be that they are transitioning, or it could be that someone, messed up in the annotation and there is a sample swap or something. So unless you have specific annotation about trans or non-binary, which is not there in these cohorts, we cannot do much about it. But I think it's super interesting, and I think people are starting to think about this, yes.
In the next trends are like more people are becoming non-binary, and it will become very relevant to look into the gender aspect.
I agree, yes. And I think this should be already incorporated in the data collection, these questions, yes. Because it's by choice, we cannot know, right? I mean, the sex, even if it's not there, you can infer from the data. The gender is cultural, so yeah, we need to add this information in the, but I mean, sometimes not even the sex is in the I mean, sometimes it's not even in the databases. Imagine gender, or they get mixed. I mean, yeah, it's tough, but we need to start thinking about this earlier in the data collection setup. Yeah, but it's very important.
And one methodological question is a bit related to that. So in the NIH grant that you presented at the end, so it seems clear that data are really longitudinal. But as per the other data, I'm wondering, are there real longitudinal data?
No.
Or just cross-sectional data at different...
None of them is longitudinal.
At different times?
None of them is longitudinal, but right. Because they are the organs.
So when you look at aging, actually, when you try to interpret trajectories, it's cross-sectional.
Yes.
So actually, you might have, depending on the generation of people, so gender-specific exposomes that would be different in people who are maybe 80 years old now, that is totally different from people who are 30 years old now, right?
I mean, yeah, the environment we cannot control. I mean, we have information about the smoking and other things, but I agree.
Yeah.
I mean, these were selected in a 5-year period, but it's true that we have different ages, so yeah, of course, the exposome is very different.
Yeah, I know that there are specific population cohorts. So, for example, Twins UK
Yes,
and Northern Finland birth cohort.
yes.
So we have quite an extensive program of questionnaire with longitudinal follow-up. It's not the longitudinal follow-up, at least in Northern Finland birth cohort, is not super exhaustive. You have 2, maximum 3 time points, but it's already something.
So this is great.
And with
but
questionnaires, and you might also have genetic data and epigenetic data.
But you don't have what's happening in the tissues.
No, you don't have.
So, I mean, it's very cool to have longitudinal data and way more powerful than cross-sectional. I absolutely agree. But it's a huge limitation where we are interested in understanding what's happening at the tissue level.
Yeah, right.
For the immune system, yes, it's great. But yeah, for the rest, yeah.
Okay, I just want to come back a little bit to the non-binary, because partially non-binary is also a phenotypic expression independent of hormone supplements. And then you do a binary analysis. So likely the phenotype is 2 Gaussians with some overlap in some ways. So when you do a non-binary analysis, but rather a kind of clustering or whatever analysis where you look at what is the potential overlap, did you ever look at this?
Wait, so the biological sex is binary.
No, the chromosomes are binary, but the
biological
gene
sex?
expression.
No.
Yeah, well, it depends on what you define.
But what we do is we take biological sex and we analyze this. The information on whether each of these donors is countering as non-binary men or women, we don't have it.
No, but you have a lot of information where likely when you do clustering analysis,
Yes.
it's not going to be 2 totally
No,
separate
of course,
clusters.
sex,
So there will be
yes,
some
no.
overlap almost for
none
sure.
of the phenotypes that I presented separate. I mean, if you look at genetic ancestry, you don't separate. The proportion of the variation in gene expression explained by sex is very small, yes. So there is some overlap. But whether the people that are in the middle would consider themselves non-binary, I'm not sure about this.
No, I agree. I totally agree. But then the question is whether then a binary analysis is the one to do, or rather that you rather say it's like, look, we have different types of expressions, and it might be totally different. Huh? So if you take the extreme male, the extreme female in your biological sex, might be totally different than the intermediate one.
But what I think they disagree because biological sex is binary.
Well, there's also some with kind of multiple chromosomes. Huh?
These
So it's not completely
They are
true.
not. These are
Okay.
conditions.
Okay,
Yes.
yeah.
These are not healthy donors. Yeah.
Yeah.
But they will agree that there is intersex and they...
No, I agree. But these are people that are considered like that they have a condition.
I think if you talk to them, I think we need to be careful here, because if we talk to them, like they play in the Olympics and they are not, they don't have a condition in the sense that it's impeding their ability. They are like
No,
that.
you're right. Well, condition, but it's a condition.
Maybe it's
It's
an
not a
outlier.
disease, it's a condition, no? Or no?
Maybe it's an outlier in your methodology that you say we're going to exclude these people because they don't
Well,
conform.
I don't exclude them, eh? I mean, the people from the databases exclude them.
Well, you can say, well, I mean, from my perspective, sex is not binary either. But
No?
you can. It depends how you annotate. But we can discuss.
I
it
thought
for hours.
No, but-
that was not under debate. Are you serious? That you don't think it's binary?
Well, there is a proportion of people that you can claim
Yeah.
biological sex is one or the other, and they have to choose or cut or not. They used to do it at the beginning. Now they wait. So.
Yeah, okay.
No, this is exactly why a continuous analysis sometimes is interesting, and you have the data. I do agree that these methodologies are a lot more difficult.
No, but this is not an excuse. No, I mean, if it's that difficult in a methodology because it's more complex, but we need to take, I agree, diversity into account. This should not be a reason. No, it's more complex. I mean, this is the reason why people excluded females or you know, people from our ancestry. So I agree, this should not be an argument. No, it's more hard. Well, we just figure it out.
Yeah.
No, but no, I mean, in the limitations, I think we do say it. But in my, yeah, okay. I'll think about it. Yes.
It sounds like a good topic for Friday morning, where we'll be talking about digital bioethics. So I guess you should come.
Other questions? I want to come back to your data set. I mean, it seems you have a super, super interesting data set, but how do you get so many samples of tissues in individuals? Is this in living donors?
No. This is an NIH project from the US. It's a huge project. It's not, I mean, I'm part of the consortium, but it's a tremendous effort, and these are all dead donors.
Yeah, but they died for a specific reason, no? So how much is it biased?
Well, mostly car accidents.
Okay.
So
they do a selection that the pathology is as limited as possible.
That they don't have cancer. They do have some conditions, like I've shown, like diabetes and some cardiovascular conditions, but they are not, they don't, I mean, some of them died of heart attack, I have to confess. But we try to get a general population to look at healthy donors. As healthy as a population of, that includes 70-year-olds can get, yes.
And if you look at then sampling, for example, because whenever you take the tissue, especially also with genes, if I mean, there's too much time,
yeah, so this is a variable.
Alteration.
Yes, you're right. It's a huge batch effect. We control for that in our models, yes.
Okay.
Yes.
Other questions? Maybe given that we have a lot of students, maybe it's interesting to see is like why as a biologist or how as a biologist did you end up in the BSc and why are you in the BSc?
Well, because the kind of analysis that I do, it cannot be done in anywhere else. So, I mean, the single cell analysis of 1,000 donors, of 1 million cells and 50,000 genes, and we do this clustering and trajectory analysis, it cannot be done. I mean, we have been asking for more and more memory, or, none of the analysis that we do can be done in a regular computer, or it would take years. So that's why I'm in the...
But what's the issue then, computing or memory or storage?
Both, it's both. Because of course, I mean, we use the image analysis, and this is also very demanding.
Well, we also do image analysis, and we use a cluster, or we go to the BSc.
Right.
So in that sense, everybody can go to the BSc.
Yeah,
so what's
the
the advantage of being in an institution like this?
Well, you don't have to think about asking for resources, you just have them.
Now, the other thing is also you're a biologist, but it seems like you use a lot of computational techniques. Is that because you trained yourself in computational techniques? And then the type of people that you have in your lab is like...
I mean, the lab is extremely diverse. So I have mathematicians, biologists that started, in computational biology like I did, but with a formal training, which I didn't have. I'm self-trained. But, I mean, I did biology, and then I started a PhD in bioinformatics, and I never left. And then when I did my postdoc at Harvard, I already was a bioinformatician, and nobody knew that I had no training on computer science. And then, I mean, I fell in love with programming, but when I started studying, there were no... I mean, bioinformatics was not even a degree, so I had a question, and then they told me, you need to learn to program to answer this question, and then I learned. But now, of course, the people that I get in the lab are, of course, have, there is the degree of bioinformatics. I get people from the UPC or from the UPC Masters, so it's a very heterogeneous lab. But it's true that the questions, I don't care about the method so much. I want to answer the biological questions. And then I get the people that has the background to answer the biological questions. So the methods for me are a means to an end and not,
Yeah. This also comes back to the fact that when you have these large data sets, very often people approach this as fishing expeditions in some ways. That they say it's like, okay, we have this data, let's see if we use all these methods, what comes out.
Right.
Is that the right approach, or what approach do you think needs to be done with these data sets?
I think it's always good, and I tell my students that they need to think about what is that they are trying to answer. And when we start a project, we have a question. The questions that I post here, I had them before I started. I didn't put them a posteriori. But it's true that if you work with large data sets, you might have an unexpected result, and then you follow up on this, like what I explained in the nerve with diabetes, that was unexpected. So it's a bit of two. But if you talk to, my husband is an evolutionary ecologist, and they of course think that this is a fishing expedition still, because they, they go to the field, they plan their experiments, it's different. But I would argue that we have some hypotheses, some ideas of what should happen. But yeah, but if you work with large data, you could still do fishing expeditions and get a good paper out of it, yeah.
No, but I like the approach that you say, we have a question, you look at it at a genetic level, and then you say, okay, we see something weird, and then you go to the tissue and so on, which I think is the way to do. But so in that sense, I don't find yours pure fishing expeditions, on the contrary. But. Okay, any other questions?
When you said about the tissues that change the most depending on the traits, like for the different ancestry, you said that it was skin, but I think the data set only has Europeans and African.
Yeah, it's terrible.
Okay, so if you added more ancestries, do you think it would still be skin the most?
Yeah, that's a great question, and I don't know actually, and it's a great question. And one of the, I haven't talked about this, but one of the things that I think the community should address is that we always focus on European descent donors. It's horrible, and I have a paper that is under review now that we show that in transcriptomics, even the databases where they say this is a gene, this is not a gene, are biased towards European population. So we need to fix this. And this project at least has some African donors, but the developmental GTEx, for example, is really trying to address this, and we're going to have much more donors from different genetic backgrounds. I would like to address this question, actually, because I think it's super important. And the community needs to switch. Of course, include females, but it's not only females that we have neglected. We've neglected, everyone that is non-white. So, yeah, very good question.
Yeah, as a matter of fact, so, there is these genome-wide association studies, and during years and years and years, they have been heavily biased because they were mostly done in those populations who had then already population cohorts. So mostly Western countries and Northern Asia. And now, as people are recognizing then these limitations, so you have population cohorts so being developed a little bit more distributed in the world, so people are using then different, so they are developing in a cohort and then they are validating in another cohort. So that's becoming the best practice. So I don't know if there is a community also behind. So
I think the genomics, yeah. So there are many papers on the genomics community, so the people that look at the DNA, not the transcriptome, saying this. And now myself and other researchers are trying to say it's not the genome only, it's the epigenome, it's the transcriptome, all the layers on top of the genome are also biased. So I agree that with GWASs, now there are some African and Asian GWASs, it's moving forward, but the other omics community is lagging behind by far, like really even worse than the GWAS, the genetics community, yes. We need to fix it. But this needs to also come down to the the NIH was one of the main funders. This is the American institution that funds science. Sorry, and now, they cut all the funding to address this. Words like female banned, ancestry, genetic ancestry also forbidden. So this is, researchers, we can say we want to study this, but we need to funds to do it. So it needs to, we need to say it out loud that we need this kind of research. Yeah.
More questions? Okay, I think it was a very interesting morning. Thank you very much for coming.
Thank you, Marta.
For the discussions.